Hello, my name is Heidi Johansen-Berg. Thanks very much for the invitation to join you for this talk, and I look forward to our discussions later. So I'm going to be talking to you about our studies imaging rapid plasticity in the sensory motor system using high-field MRI. So my group is interested in brain plasticity, how the brain changes with experience. And we study plasticity in a number of different contexts. What happens to the brain when we develop, when we learn new skills, such as learning to juggle, when we change our lifestyle, for example, taking up exercise, or what happens in the brain when we suffer damage to the brain or to. And with each of these different areas, we're interested both in terms of plasticity of the brain's structure, its physical properties, and plasticity of the brain's function, changes in the way in which it works as a result of experience. And we can study both structural and functional plasticity at different levels. At a macroscopic level, we might be observing changes in the structure or function of whole brain regions, whereas at a microscopic level, we might be using different methods to be able to study changes at the level of individual neurons in the brain. And we take various different approaches to study these phenomena in different ways and at different levels. And I'm going to focus today's talk on the studies that we've done using high-field MRI to address some of our questions around brain plasticity. And the first question I'm going to discuss with you is, when does plasticity happen? Under what circumstances do we change the brain? And for this set of experiments, we're going to be focused on body maps in the brain. So the idea that the brain contains functional maps of the body is an idea that's been around for a very long time, 100 years or so. This is based on a classical drawing from Wilder Penfield, the Canadian neurosurgeon, who used electrical stimulation to stimulate the brains of his patients undergoing brain surgery to derive this very famous homunculus in the brain, the motor and sensory representation of the brain, whereby he was able to demonstrate that stimulation of different areas along the sensory motor strip evoked movements of different body parts. So we know that these maps exist in the brain. To what extent can we visualize them in intact brains who aren't undergoing brain surgery? Well, this is one application for which high-field MRI is particularly powerful, because we know that with high-field MRI we can use the additional contrast and signal that we get from our high-field magnet to gain higher spatial resolution in a shorter period of time. And one of the things that allows us to do is to derive detailed functional maps at a level of resolution that would be less feasible to achieve at conventional field strengths. So, for example, this was one of the first studies that we did with our 7 Tesla MRI scanner in Oxford when it was first installed nearly about 10 years ago now. So my student James Kolosinski used the scanner to perform a high-resolution fMRI study of individual finger representations within the human brain. So in a very simple set of experiments, participants were in the fMRI scanner and performing sequential movements of the fingers of the hand, and using a phase encoding design, James was able to record this travelling wave activity travelling up the central sulcus as this task was being performed. So those signals could then be used to derive these beautiful individual digit maps, whereby voxels are assigned to the finger with which they have the closest association in terms of when the finger was moving versus how the brain was responding. So we can derive these very clear individual digit maps at the level of individual participants. So in each participant, we can visualize these digit maps. Here's an example from 3 participants. There are other participants in the paper if you're interested. And one thing you can see straight away is that the basic topography is very consistent across those people. So in all of those individuals, we see this topographic representation with the 4 digits that are being moved mapped onto this inferior superior axis along the central sulcus, which we can show schematically there on the right hand side, where you see the maps corresponding to digits 2, 3, 4 and 5. And in this initial study, James scanned a number of different people, was able to show that the topography was consistent across people. And he also brought those individuals back for repeat scans either 24 hours or 4 weeks later. And so now what you're seeing is each row represents a different participant, and then the individual images along that row represent a different time point. And what you can see illustrated then is that the topography is consistent across people, but the fine details of the maps vary slightly from person to person. So the precise size and shape of the different clusters, for example. And yet within an individual, as we move from left to right here, we see a very clear consistency in the details over time. So within participants, the maps are highly reproducible over time. They're very stable over time. We find the same maps from one occasion to another. So therefore, this approach provides us with a useful starting point to ask the question, when do these maps change? So given they're generally stable, under what circumstances can we shift these cortical maps? So can we exploit the power of our 70 scanner to derive these fine detailed maps in order to ask how they change over time? So to ask this question then, to assess short-term plasticity in digit maps, James performed an experiment in which subjects came to the scanner and had 4 different scans on different occasions that you see illustrated here, separated by 24-hour periods, where in one 24-hour period, subjects were able to go about their business as usual, whereas in the other 24-hour period, the glued period on the right here, James glued together the index and middle finger of their right hand for 24 hours. So he used a special surgical glue which can very effectively bind the fingers together, but is then and leave the skin surfaces exposed, but is then easy to remove the following day. And the question is, does this gluing procedure, whereby 2 digits are fused together, does that alter those cortical maps in any way? So those of you who are familiar with the sort of classical literature from non-human primates might find this experiment somewhat familiar, and that there have been many studies in previous decades where the digits in non-human primates have been sutured together and then electrophysiological mapping used to monitor how that type of long-term manipulation alters digit maps. And based on that classical literature, we hypothesized the scenario that you see illustrated here. So based on those classical studies, we expected that when we glued together digits 2 and 3, the cortical representation of those 2 digits would move closer together. Interestingly, what we found was a bit different from that. What we found is illustrated here. So on the graph here, on the y-axis, you're seeing the degree of overlap between cortical maps from pairs of digits. And what we find is actually that, to our surprise, the overlap for digits 2 and 3, the glued fingers, does not change as a result of the manipulation, whereas what changes is the degree of overlap with the other remaining digits. So specifically, as you see in pink, there's less overlap for digits 3 and 4 after the gluing, and there's more overlap for digits 4 and 5 after the gluing. So what changes is the degree of overlap between the unglued fingers. So in other words, what happens is illustrated here. So as we glue together, after we glue together digits 2 and 3, we get increasing overlap between 4 and 5 and increased separation between 3 and 4, presumably because subjects are having to adapt very rapidly to how to use their hand differently and use those remaining digits 4 and 5 quite differently as a result of the gluing manipulation. So this was intriguing, and we considered that that neural change that we were detecting with our high-resolution fMRI would predict specific patterns of behavioural change. So specifically, if what's happening in the brain is what we've observed in our fMRI data, which is greater cortical overlap between 4 and 5 and reduced overlap between 3 and 4, we might make clear behavioural predictions as a consequence of that. We might predict that digits 4 and 5, which are now more overlapping in terms of their cortical maps, subjects should be worse at discriminating tactile inputs to those digits 4 and 5. Because, if you like, there's more confusion in the signals from those digits. Whereas for digits 3 and 4, whose cortical maps have got further apart as a result of the gluing, subjects might get better at discriminating tactile inputs to those digits because they're easier to separate in cortical terms. So what James did was perform a follow-up experiment with behavioural outcome measures in the same time points and gluing manipulations to test these behavioural predictions. And what we found was that the behavioural change indeed fits the prediction from the neural change. So now what you're seeing on the graph is behavioural data. So temporal order judgment task, which is a simple psychophysical task where subjects are presented with pairs of tactile stimuli to 2 digits, and they have to say which finger was stimulated first. And you vary the temporal interval between the stimuli, and you can produce a nice psychophysical curve from which you can quantify how good each individual subject is at differentiating each pair of digits. So that's the behavioural measure that's being plotted on the y-axis, change in that measure. And you can see that again, all of the change is happening after the gluing period of time, as we would expect. And again, that the direction of change for each of those digit pairs indeed fits with those predictions. So in other words, the pink, for the pink pair, which is digits 3 and 4, where we saw less overlap from the fMRI data, subjects did get better at discriminating tactile inputs to those fingers, whereas for the blue digit pair, 4 and 5, where we saw more overlap on the fMRI data, subjects indeed get worse at discriminating tactile inputs to those digits. So what this study shows then is that we can exploit the high spatial resolution offered by 7T fMRI to derive these very detailed digit maps, cortical representation maps at the level of individual subjects, so that we can then be sensitive to conditions under which these detailed maps can change as a result of experience. So in this experiment then, we have a very short-term change in experience, just 24 hours of altered experience. And we see that, interestingly, what seems to change is the way in which the subjects use their other digits to compensate, if you like, for the glued digits. But this is a very short-term, artificially induced change in sensory motor experience. What happens with much longer-term changes in sensory motor experience? Well, we've studied this in the context of upper limb amputation. And this is work that was led by Tamar Makin when she was a postdoctoral fellow in the lab, and she's now a professor at UCL. And Tamar brought to the lab an interest in amputation, which we combined with our interest in plasticity to explore the ways in which the brain changes or adapts as a result of limb amputation. So you may well be familiar with the sort of prevailing model of how we think about limb amputation, which is this idea that if we consider again this the body map, the Penfield's body map in the sensory motor strip that we are very familiar with, if we remove the inputs from a body part, let's say the hand, through amputation, that then means the inputs to that area of cortex are missing. What's thought is that what happens is that neighboring cortical representations, the face, for example, invade that freed up cortical real estate and they take over the deprived cortex. So this is how scientists have thought about reorganization following amputation for a long time, and there is some evidence in support of this idea. But this model is very much focused on sensory deprivation, the loss of input from the missing limb. And yet we know that many individuals with acquired limb amputation, so amputation, they're born with 2 arms, for example, and then one is amputated at some point post-infancy, we know that many of those individuals experience what we call phantom limb sensations. So although these are no longer there, the individual has very vivid sensations of the arm. They can feel the arm, they can voluntarily move the arm. This phantom limb is very present in their brain and in their perception, even if it's not physically present. So our question then was, well, how is that phantom limb represented in the brain? So to investigate this question, Tamar studied a group of amputees with phantom limb sensation, and she asked those individuals to move their phantom voluntarily in the fMRI scanner in the same way as we could ask intact two-handed individuals to do. And what we found, remarkably, was that there was no difference in brain activity patterns between a two-handed person moving their hand and an amputee moving their phantom hand. And you can see that by eye in these inflated cortical maps of the sensory motor cortex. And you can also see that quantitatively here when we use an unbiased region of interest to quantify the degree of movement-related activity for amputees in red and for two-handed controls in purple. Importantly, the amputees voluntarily moving their phantom limb is not the same as imagining moving the phantom limb. So there's different signals when they're moving the limb versus simply imagining moving the limb, the same way there would be in somebody with an intact limb. And you can also see here a control in green, which is one-handed individuals who were born with only one upper limb, who typically have phantom limb. And when they're asked to move the phantom, to move their missing limb, which they don't, which they aren't typically able to do, but they imagine moving the missing limb, you can see that manipulation does not activate the brain in the same way. So where there's a phantom present in the brain, that phantom limb is represented just as strongly as an intact limb. So these data were acquired at 3T using a conventional research field strength. But having observed these data, we then asked the question, well, how organized is that representation of the phantom? This limb, this hand, may have been missing for many, many decades. To what extent is the detailed intrinsic functional organization that we saw in our previous study with the digit maps, to what extent can that organization be preserved even in the absence of the limb? So to ask this question, then we ran a study at 70 where we could derive those individual digit maps like we were just seeing. And Sonny Kickett, who was a PhD student in the lab at the time, performed a study in controls and amputees. You're seeing example data from 2 controls here, where you're seeing a similar representation of digit maps to what we saw in the previous study. Now they're moving all 5 digits, so we have 5 different colors, but again, you're seeing the same topographic gradient moving up the central sulcus of digits 1, 2, 3, 4, and 5. So a group of controls were studied, and that detailed digit topography was found, just like in the previous study. But then Sane was able to find 2 individuals with acquired limb amputation who were safe to go in the 7T scanner, which is a challenge in itself because many of these individuals have experienced the amputation through a traumatic accident, which might then make them not safe for scanning at high field. But she found 2 who were suitable for 7T scanning and who reported they were able to voluntarily control the individual digits of their missing phantom limb. And what we found remarkably was that the topographic mapping in those individuals was just as detailed, just as organized, and just as clear as it was in the control subjects. So in the paper, we have various ways in which we quantify the topography of these functional maps and find that they're just as organized in the amputees as they are in the. So with the 7T scanner, then we were able to reveal preserved fine-grained digit topography despite decades of limb loss. So to summarize then an answer to that first question, when does plasticity happen? We've shown using 7T that short-term change in experience, just 24 hours of gluing 2 of the digits together, evokes rapid remapping of functional brain maps. But nevertheless, the basic topography of brain maps is remarkably resilient to altered experience, as we could see from our amputees with preserved digit maps. So for the second half of the talk, I want to focus on a question that we've addressed in the lab: where does plasticity happen? And we're thinking here in terms of where within the brain, in which location within the tissue does plasticity happen. So we'll all be familiar with the idea of synaptic plasticity. That is the prototype type of plasticity in the brain. It's very well described. Synaptic plasticity as a result of experience, Hebbian processes such as long-term potentiation and long-term depressant underlie learning and memory. That's a very, very clear phenomenon that we understand incredibly well. We've been interested in asking whether there are other types of plasticity beyond the synapse that might complement synaptic plasticity, and in particular, whether the myelinated axons offer an alternative site for plasticity in the brain. So, as I'm sure you're all aware, axons in the brain are surrounded, or some of them are surrounded by a myelin sheath, an insulating fatty substance that provides support to the axon and allows rapid signal transduction. And some of our brain imaging methods are sensitive to variation in myelin as well as other features of the white matter structure. So to address this question then of where does plasticity happen, we've carried out a number of studies using brain imaging methods. So here's just one example of these studies where we showed here using 3T MRI that acquiring a novel visuomotor skill in healthy young adults, in this case learning to juggle, changes brain structure. And it not only changes the structure of the grey matter in the cortex, but also changes the underlying white matter structure. So here, for example, you can see that when we've compared brain scans taken before and after a 6-week period of training on a juggling task, we can use voxel-based morphometry to detect images in grey matter volumes, shown here in red. And interestingly, these areas of increase are in areas of the medial occipital and parietal cortex that we know to be particularly involved in reaching, grasping for objects in the periphery of visual space. So a function that's very relevant to the particular skill being trained here. But then interestingly, in addition to those cortical changes, we're seeing, as shown here in blue, changes in the structure of the underlying white matter tissue where the axons would be located. And what we're detecting is using diffusion imaging, we're finding an increase in FA, or fractional anisotropy, which many of you will know is a metric that is sensitive to this directional dependence of water diffusion in the tissue. And this is a useful thing to measure in the context of white matter, because we know that in tissue with highly directionally oriented structure, like a white matter fiber bundle, water diffuses more easily along the axis of the bundle than it does across the axis of the bundle. So there will be a high degree of directional dependence to that diffusion, and that directional dependence will be captured by this metric of fractional anisotropy, or FA. And we know that FA varies with various different features of the tissue, including, for example, the membrane integrity, the myelin integrity, the axon diameter, etc. So this study showed us that something changes in the white matter structure as a consequence of training. The question is what changes? How should we interpret that structural change in the white matter that we've detected? Well, a challenge here is that the metric that we're using of diffusion fractional anisotropy is not a specific metric. So it's an indirect measure, it's an indirect probe of tissue structure. It's telling us about the directional dependence of water diffusion, which is a metric that is sensitive to a number of different lying scenarios. So some of those are illustrated here. So as we detect an increase in FA moving to the top from the bottom of this slide, that change could reflect a number of different underlying scenarios at the level of the axons in the cells. So moving from left to right, it could reflect an increase in the packing density of the axons, a decrease in the diameter of the axons, an increase in the organization or coherence of the bundle, or, as on the right, could reflect changes in the myelin. It could reflect formation of new myelin on previously unmyelinated axons, or it could reflect a thickening of myelin from previously myelinated axons. Any one of these scenarios, or a combination of these scenarios, could underlie the change that we've seen in our imaging measure. And we can't tease these apart using imaging alone. So one thing that we have done in my lab for the last 10 years or so is have a parallel set of experiments that we run in rodents where we can directly measure and manipulate some of these phenomena more specifically than we're able to do in our human studies. So here, for example, in some work from Cassandra Sampaio-Baptista from when she was a PhD student in the lab, she's now a lecturer in the University of Glasgow in Scotland. She trained rats on a motor learning task and then scanned their brains using diffusion imaging post-mortem and compared trained rats to various control groups and was able to show, again, an increase in FA, shown here by this red cluster, in white matter underlying the motor cortex in the trained hemisphere in these rats. So this, if you like, is the rodent equivalent of the juggling result that we found in humans. But then because this is an experiment in animals, we can follow up and do measurements and manipulations that wouldn't be possible in humans. For example, here, immunohistochemistry, staining for myelin basic protein, where we're able to show that in the same area of the white matter, there's increased myelin density in trained animals compared to control animals, suggesting that indeed some of the changes that we're detecting with our imaging measures do indeed appear to reflect myelin plasticity. And we've subsequently done various studies with other types of MRI metrics that are more specific to myelin and found similar results. And we've gone in and manipulated the process of myelin plasticity through transgenic manipulations and found that that does indeed impair the animal's ability to learn new motor tasks. But I won't go into those data here in order to not get too far off topic from high field MRI. I just wanted to tell you the story of these imaging studies have shown that myelin plasticity may be important for learning. One of the many outstanding questions is how quickly can this type of activity-dependent myelination occur? And this figure is taken from a study from the US, where Gibson and colleagues used optogenetic stimulation in rodent brains to drive neurons in one of the motor areas of the rodent brain in order to see what effect that stimulation would have on interconnecting brain pathways. And what they found, as illustrated in the graph on the right, was a rapid proliferation of oligodendrocyte precursors, so the myelin-making cells in the brain, occurring very quickly after just half an hour of this type of optogenetic stimulation. So raising the possibility that even fairly brief periods of activity modulation could result in myelin plasticity. So obviously this is a very artificially induced activity modulation of directly stimulating the neurons, but very specifically and precisely. We've been interested in studying some of these questions in the human brain using a much less precise but non-invasive method of modulating activity, in this case with 7T neurofeedback. So the idea of the neurofeedback experiment is that you tap into the individual's own capacity to control their own brain activity through feedback. So in these examples where it's fMRI neurofeedback, we would have somebody lying in the fMRI scanner performing a particular task. We are continually reading out their bold signal over time, feeding that into a computer, which is doing a rough and ready quick analyses of those data, which is being updated ER. And then we're feeding back a visual representation of that activity, for example, a moving bar on a screen, where that visual representation represents their brain activity. the subject is instructed to manipulate that visual representation in some way, for example, increase the height of a bar on the screen, which corresponds to manipulate their brain activity in a targeted way. And it turns out that this type of feedback, which has now been used in many, many different experiments with many different brain regions, subjects are able to use this type of feedback to manipulate their brain activity in particular ways. We've been interested to study this approach of fMRI neurofeedback in the motor system to understand whether we could use feedback to modulate brain activity. And we're partly interested in this as a research tool, but partly as a potential future application in the domain of stroke rehabilitation. So there is interest in neurofeedback as a clinical tool because for a given clinical problem, if we know what the optimal pattern of brain activity is, then perhaps we can use neurofeedback to drive the brain into that more optimal pattern of activity and therefore improve outcomes for patients. So, but this first experiment I'm going to talk to you about is in healthy people, where we're purely using neurofeedback as a research tool. And so we're using neurofeedback in this experiment to modulate sensory motor activity in order to determine whether modulating activity changes brain structure. So our previous experiment showed that long-term manipulation of behavior, so a training over 6 weeks, changed white matter structure in the human experiment, or training on a motor task over 2 weeks in the rodents changed white matter structure. Here we're interested to see whether just half an hour of neurofeedback-based activity modulation changes brain structure in healthy humans. So for this experiment then, which is again by Cassandra Sampal-Baptista, subjects came into the lab for 4 different scans, and they experienced 2 different neurofeedback conditions within those scans. It's a very simple neurofeedback experiment in which our feedback regions of interest are centered, as you see here, on the sensory motor cortices of the 2 hemispheres. And so the degree of movement-related activity from those 2 regions of interest is fed back to subjects through the visual displays that you see on the right there, where the height of the red bar represents the degree of movement-related activity for the contralateral motor cortex, and the blue bar for the ipsilateral motor cortex. Subjects are simply instructed to move their hand while observing the moving bars on the screen, and they have 2 different neurofeedback conditions. So on the left, the association condition, they're instructed to try to increase the size of both of those bars, whereas for the dissociation condition on the right, they're instructed to try to decrease the size of the blue bar while increasing the size of the red bar. So what that means then is that in both neurofeedback conditions, they're trying to drive up the red bar, which is the contralateral motor cortex activity, whereas what differs between the 2 conditions is what they're trying to do with the blue bar, which is the ipsilateral motor cortex activity. In one, they're trying to drive up the ipsilateral activity, whereas in the other, they're trying to drive down the ipsilateral activity. And if you look at the fMRI data, so this is now a 7T fMRI study, we can see that indeed they are able to use that neurofeedback to control their brain activity in the instructed direction. So here what you're seeing is the fMRI activity from the ipsilateral region of interest in blue and from the contralateral region of interest in red, where the data from the association condition, the IPSI up condition, is shown by the white bars, and the IPSI down condition is shown by the grey bars. And this is over the 3 blocks of neurofeedback that they experience on each day. So on the right-hand side, for the contralateral hemisphere, you can see that they're keeping that activity very high, as you would expect with the contralateral sensory motor activity. It's kept high and it remains high throughout and doesn't differ between those 2 conditions, both of which are trying to drive up the contralateral hemisphere. Whereas in blue, for the ipsilateral motor cortex, you can see that clear difference between the 2 conditions, and that they are being able to follow the instructions such that the IPSI up condition in white is increasing, whereas the IPSI down condition in grey is decreasing. So they're able to use the feedback to modulate their brain activity, and in particular to modulate the activity of the ipsilateral sensory motor cortex. So then our question is, does that modulation of activity alter their white matter structure? So we acquired DTI scans before and 24 hours after each of those neurofeedback conditions so that we could look for changes in white matter structure as a result of neurofeedback that differ between those two neurofeedback conditions. And when we ask that question, so the group by time action, in other words, we find a cluster of significant voxels here shown in red on the left, where there's a change in corpus callosum anisotropy, which is detected 24 hours after that change in activity. So we see this interaction, and if we look at the data that are driving that interaction, you can see that unpacked on the right there. You can see that what we're detecting within that cluster is an increase in FA for the increased IPSI condition and a decrease in FA for the decreased IPSI condition, such that we see a significant change between the two. So what this suggests then is that modulating activity in a targeted brain area, in this case the ipsilateral motor cortex, results in a change in white matter structure within just 24 hours. So obviously based on the FA change by itself, we can't be sure what's driving that change, whether it's a myelin plasticity effect or whether some other feature of the white matter is changing. But we can see that after even this short-term change in activity, we can change the structure of those pathways, which suggests that maybe through a neurofeedback training, we can not only alter activity during the feedback itself, but also change the brain in such a way that might be relevant for future activity. So therefore, then that gives weight to the idea that neurofeedback might be relevant as a clinical intervention for recovery and rehabilitation, because it appears to be changing the brain. So if we can change the brain in a desired direction, maybe that would have beneficial clinical outcomes for patients. So we've this possibility. I'm going to talk just very briefly about this clinical trial that we've conducted in chronic stroke patients. This was quite an intensive trial over a number of months and patients with a number of different measurements for patients. So we performed pre and post MRI measurements and neurofeedback training on 3 days within the intervention week, and then various clinical and EEG outcome measures, as well as a long-term clinical follow-up. Just to highlight a couple of aspects of the results, and you can see the paper for more detailed reporting of the results of the trial. So this, as I said, was an experiment, was a study in chronic stroke patients who had unilateral movement impairment in their affected limb. And what we're trying to do is similar to the experiment in healthy controls, is shift the laterality of activity. So all of these patients in the real neurofeedback group had the same kind of instruction. They're instructed to drive up the activity in the contralateral motor cortex and drive down the activity in the ipsilateral motor cortex, the result of which is to increase the laterality of their activity to make it more lateralized towards the stroke affected hemisphere. And there's a lot of evidence to suggest that more lateralized activity after stroke is associated with better clinical outcomes. So we're trying to use the feedback to drive the activity towards a more lateralized pattern. What we find, as in a number of other studies, is that patients are able to use the feedback to drive the activity in that instructed direction. But on the whole, they tend to revert back to baseline once the feedback is removed. However, in this experiment, where there were repeated sessions of neurofeedback over a number of different days, we did start to see some evidence for transfer of those neurofeedback changes to other tasks once the feedback is removed. And in particular, we saw variability across participants in the degree to which that transfer is observed. So it's very common in neurofeedback experiments that there's a high degree of individual differences in how well people are able to use neurofeedback, and the same was true in our clinical study. People varied in how well they could use the neurofeedback, and that was quantified here by a neurofeedback success index. And what we found was that that success index correlated with the amount of transfer of those neurofeedback effects. So responders, people who could use the neurofeedback very effectively, showed the greatest transfer effects. So that on a new motor task, they produced more lateralized activity as a result of the neurofeedback. So some evidence that if we were able to identify responders, then perhaps we could drive positive effects of that neurofeedback training. But interestingly, coming back to the question of white matter structural plasticity, we were able to show at the group level that the real neurofeedback group had a structural change, a structural white matter change as a result of that neurofeedback. So specifically, they showed increased symmetry of FA within the corticospinal tracts with the real neurofeedback. So what we've done here is identify regions of interest in the corticospinal tract, quantified FA asymmetry. At baseline, all of the patients show asymmetrical FA, reflecting reduced FA in the affected hemisphere compared to the unaffected hemisphere. But with feedback, that improves in the real group and not in the sham group. So in this experiment, the sham control group receive somebody else's feedback. They're yoked to a real participant. So we see, I have a typo here, it's decreased FA asymmetry in the real neurofeedback group compared to the sham neurofeedback group. And that's driven by increasing FA in the stroke affected hemisphere. So just like in the experiment with healthy controls, it seems that the activity modulation can drive brain structural changes. So that's promising in the context of a clinical intervention, because even in the absence of clear-cut behavioral effects at this time point, if we're driving brain change in a desired direction, that might provide a good substrate for future functional gains. And in this experiment, even though our primary clinical outcomes don't show any benefit as a result of neurofeedback, we did see some tentative benefits in movement times for a timed test of motor function in the real group compared to the sham group. So to summarize then, I've shown you a number of examples of how we can exploit the opportunity of high field MRI to provide us with high sensitivity to both structural and functional plasticity. So specifically with our fMRI experiments, we were able to show rapid plasticity of digit map overlap after short-term altered experience, just 24 hours of gluing the fingers together. But we were also able to show remarkably preserved digit topography many years after limb amputation. And with our structural imaging experiments, we were able to show a rapid change in white matter microstructure after activity modulation. And finally, that this might be relevant for recovery. It might provide a useful substrate for future clinical gains. So finally, I'd just like to thank my primary funders, the Wellcome Trust, all of my colleagues at the Wellcome Center for Integrative Neuroimaging in Oxford, and my research group in particular, whose work I've been able to share with you today. And I very much look forward to our discussions about these topics in the future. And thank you for your attention.